Welcome to section 8.3, where we're going to wrap up the idea of cellular respiration. So we've already talked about photosynthesis, and photosynthesis is kind of like the precursor in many ways of cellular respiration, because photosynthesis produced for us the organic molecules, which we're going to say sugar in this case, glucose, and it produced for us oxygen. And we're going to use this sugar or any other macromolecule for the most part, as well as oxygen, during cellular respiration to produce CO2, H2O. Remember, that's what photosynthesis needed. So these guys are kind of like a cycle. You know, the reactants of one are the products of the other, and the products of one are the reactants of the other. So you're going to use this to get these products, which can then be used for photosynthesis. But during cellular respiration, we produce one critical thing that's essential for life, and that's ATP. We've talked about how there's energy stored in these organic molecules, that it takes energy to produce them. Well, our bodies extract that energy during cellular respiration to produce ATP. And ATP is what all the energy requiring processes in our body use to function. So it's essential that we have a lot of ATP. We're talking thousands of ATP per second in most of our cells. So you should know the equation for photosynthesis and cellular respiration, and you should be able to memorize those pretty easy because they're opposite each other. And then we're going to discuss two distinct parts of cellular respiration. The first one, glycolysis, is something all cells will do. So this is going to be kind of a universal. And then the second part's going to vary based upon if we have oxygen. If we have oxygen, the preferred method is to do aerobic. If there is no oxygen, we will do anaerobic, which involves a step called fermentation instead. So this will be an either or. You don't do both, you do one or the other. Now the universal step in cellular respiration is glycolysis. This occurs in the cytoplasm. It needs nothing fancy. It doesn't need oxygen. It doesn't need a mitochondria. It's just going to take glucose, which is a 6-carbon molecule. Each little green sphere here simulates a carbon. And it's going to split that ultimately into 2 3-carbon pyruvate molecules. Now this may not seem like much, but as you can tell, it involves many steps, and it gives us some vital byproducts. And these other products that we get are 2 ATP, so that's the energy guy we talked about, and it also gives us 2 NADHs. Now NADH is a lot like NADPH from photosynthesis. I always remember NADPH, the P is for photosynthesis. But it's very similar in that it's going to store electrons. It's an electron carrier, and those electrons have energy that we can extract later to make more ATP. So NADH you can think of as like potential ATP if we have the right equipment. So we can't use it yet, but it's still useful later on. Now after glycolysis, we're going to take our pyruvate and we're going to move inside the very centermost part of the mitochondria called the mitochondrial matrix. And that's where the Krebs cycle is going to take place. Now the Krebs cycle effectively has 2 parts. There's a pre-step, one that occurs before the Krebs cycle, where we convert pyruvate to acetyl-CoA. Basically you go from a 3-carbon pyruvate to a 2-carbon acetyl-CoA. In the process we make 2 NADHs, which is kind of useful for us. So this acetyl-CoA that we got from the conversion step is going to go into the actual Krebs cycle. And during the Krebs cycle, the acetyl-CoA is going to get ripped down into CO2, which has effectively had as much energy as possible extracted from it. So we're kind of done with our sugar at this point. We've broken our sugar down into CO2, which we can then exhale as a waste product. Now in the process of this acetyl-CoA to CO2 conversion though, the Krebs cycle, we're going to make 6 NADHs and 2 FADH2s. These are also electron carriers, right? Just like we talked about before. The FADH2 just happens to be one that's not quite as good as NADH, but we still make some of it, so that's still cool. We can still get some energy from it. We also will produce 2 ATP, so very little energy is produced, but a little bit is directly. Most of our energy is going to be stored in these 8 NADHs total, and these 2 FADH2s that we've managed to create. The problem here is we still need an extra step to extract the energy that's stored in the electrons on those electron carriers. So it's only potential ATP at this point still. But luckily for us, there's one more step. So if you look at a mitochondria, you have the outer membrane, and then you've got this folded up inner membrane called the cristae. And it's embedded in this inner membrane that we have the electron transport system or the electron transport chain or just electron transport, whatever you want to call it. So if you see electron transport or ETC or ETS, same thing. It's a series of these proteins called cytochromes that are embedded in this membrane, and it's going to function almost exactly like photosystem 2 did, where we're going to take the electrons, not from pigments this time, but we're going to take the electrons that NADH has and FADH2 has, and we're going to strip them off by converting them back to their uncharged forms. So NADH will become NAD+, because we remove an H+ and 2 electrons from it. And FADH2 is going to become FAD because we remove 2 electrons and 2 H+ from it. So we're going to kind of strip them of their electrons and a few H+. And in the process now, we can put these electrons on this series of electron transport molecules. And these electron transport molecules that are embedded are going to pass them along from one to the next as the electrons move through. And the whole purpose of this is they're going to use the electrons' energy to pump H+'s out. So in this case, they're pumping it across and out of the cristae. So we now get a gradient where there's a lot of H+ that's present in what's called the intermembrane space, this area here, and there's not much H+ on the inside. This gradient is a source of energy. So the fact that we've built this gradient now means that we've got energy stored here, and by allowing those H+'s to diffuse back in, this will be a form of facilitated diffusion, they're going to go through a protein called ATP synthase that's embedded in the cristae, and by going through there, they're going to provide the energy needed to make ATP. So this is where we're going to produce most of the ATP that we get from cellular respiration will come from this final step where we strip electrons from NADH and FADH2, use those electrons' energy to make the H+ gradient, and then allow the H+ gradient to diffuse through ATP synthase. This nets us 34 ATP. So that's 34 ATP out of a total max of 38 that can be produced per glucose. If we're doing anaerobic respiration, if we don't do this whole process of glycolysis, Krebs, and the ETC, we can only make 2. So you can see this is much, much, much more efficient per glucose. If you're selling something, you'd much rather get $38 for it rather than 2. And that's why it's so important we have oxygen. Because if you notice down here, if we put somebody on the electron transport system, at some point this electron needs to get off of it. It needs to be removed so the next electron can go through it. It's a lot like if you have a slide, someone goes down the slide. If they just sat there, nobody else could use it. It'd block it up. It'd now become unusable. So we need someone to get that person off the electron transport system or off the slide in our analogy. And that person is oxygen. So oxygen will grab some of these H+'s that are hanging around, as well as the electrons from the ETC, and it's going to use those to make water. So if we don't have oxygen, the electron transport chain falls apart, which means we won't do the Krebs cycle and we won't do the electron transport chain. These only function if oxygen's present. This is why oxygen's so important to this process. So speaking of if oxygen's not present, anaerobic respiration is literally when there is not oxygen used. And so if there's no O2, we've said the Krebs cycle becomes useless. It's making a bunch of molecules that are only useful if there's oxygen, because we have to have oxygen to use the ETC. And the ETC is essential to convert NADH back to NAD+. We need to essentially use up the NADH. And so if there's no oxygen, we can still get energy. But what we're going to do is we're going to get our 2 ATP from glycolysis. You can see those here. So we've now got our max 2 ATP. But our problem is we're converting our NAD+ to NADH. So we're using up our NAD+. And there's nothing within glycolysis or after that's going to use up this NADH. So we start to have too much NADH that we can't use because there's no oxygen. So what we do instead is we have another process called fermentation. And fermentation has 2 possible outcomes. The first is we convert this pyruvate that we produced with glycolysis to something called lactic acid or lactate. Right? In the process of doing this, the only thing we don't get any ATP. No ATP is made during fermentation. But we do end up doing one critically beneficial thing. We convert our NADH back to NAD+, which allows for it to go up and allow glycolysis to continue to occur so we can keep getting those 2 ATP. Ethanol or ethyl alcohol fermentation is very similar. Same general thing. We're going to convert NADH to NAD+. which can then allow glycolysis to continue. So it's just going to be converting pyruvate to something else to use up that NADH. We still only get 2 ATP though, so it's incredibly inefficient. This is why you will not see large organisms like vertebrates, like multicellular organisms, using anaerobic respiration. It's so inefficient, you just would not be able to eat enough and get rid of enough waste to make this work. Now we can do it briefly. So if you're jogging, for instance, you have enough ATP on hand and enough oxygen on hand to probably go for about a minute. But after that, you start to run low on oxygen, and so you'll start to do fermentation. You'll start to convert some of your pyruvates. Instead of going through the Krebs cycle and the ETC, they'll do lactic acid fermentation. They don't do ethyl alcohol fermentation, or else it'd be a very important accomplishment if you finished a marathon, because by that point you'd be wasted. So instead, we'll just produce lactic acid, and this can allow us for brief periods to do anaerobic respiration, to operate without oxygen. This is critical so you're not like running from a bear or something, and then a minute in, you just drop down. But ultimately, it's not going to be our default. We need to have plenty of oxygen for us to last long term because, yet again, it's so inefficient. 2 ATP instead of 38 ATP, and that's from the same starting glucose.